Hello students, welcome to LEELA'S tutorial. So in last class we have discussed about plant tissue culture media and types of culture, right? So some of you asked me what is the difference between differentiation and redifferentiation. So let us discuss that. Normally we are going to take the explant, that is any part of the plant body, and then we are going to inoculate in the media so that we get a callus, right? So what do I mean by callus? Callus is an undifferentiated, unorganized mass of cells, right? So when you take the explant, it can be the leaf, right? If you are taking any portion of the leaf, right, that portion is referred as your explant. So this explant, when you are going to inoculate in the plant tissue culture media and incubate under favorable conditions, what happens here? Those cells will take up the nutrients from the tissue culture media and they start dividing. So the group of cells which are unorganized mass, right? So these are off-white in color, and you can see in the culture tube you can see an unorganized mass of cells. This is the callus. When you are inoculating an explant and incubating it, the cells divide, right, to form a clone of cells, right? So now these cells, a group of cells, are referred as undifferentiated mass of cells, right? That is callus. Why you are calling it as undifferentiated mass of cells? Because they do not have any structure or function. Okay? See, normally here this leaf is an differentiated cell. That means the leaf is having structure and also function, right? The cells present in the leaf are having certain function and also the structure. The chlorophyll is having its own structure, and what is the function? Here it is photosynthesis. So here it is an differentiated tissue which you are using as an explant. When you are inoculating in a plant tissue culture media, the explant cells divide, right? They divide to form a group of cells which are off-white. Here the explant is green, right? The leaves are green in color, whereas here the callus is off-white in color because they don't have any function. It is not that the chlorophyll or any function will be attained. Here they are losing its structure and also function. Only thing is they are dividing. So now these cells are referred as undifferentiated mass of cells. They do not have any structure or function. So when you incubate further, right? So now what is the use of this callus? You have selected some plant, you have selected the explant and the media, then you have now the callus. Callus consists of hundreds or thousands of cells. Now from each cell you have to produce a plantlet by means of micropropagation, right? So that is our motto. In order to produce a clone of cells, all the plantlets which are formed from this group of cells will be a clone. They all have same phenotypic and the genotypic characters. So here once the callus is formed, when you further incubate, right, when you further culture, what happens? They become differentiated cells. Differentiated, again I said differentiated means which have their structure and also the function. Okay, so here you can see here the division ability is lost, whereas in undifferentiated cells the division ability is present but structure and function is absent. Whereas here in the differentiated cells they attain the structure and function but lose the dividing ability. Is that clear? So here you can see the specialized cells with structure and function. Now if I want to produce a shoot system from the callus, so I will produce a shoot by increasing the auxin to cytokinin ratio, right? So here the shoot is formed. That means what? The callus or the undifferentiated cell, when it is attaining a shoot system or transforming into a shoot, that means it has got some structure and also it has got the function. In case if I want this callus to produce the roots, then again it will be having some other function and other structure, right? It may be any cell which is produced from the callus but once it is getting differentiated, it will lose the dividing ability and it will attain structure and function. Is that clear? So next, further incubation, here you are getting dedifferentiated cells. See here, it is undifferentiated, differentiated, now it is dedifferentiated. So here in the dedifferentiated cells, they regain the division ability and lose the function. Okay, so here what happens once the cells are matured? If you take, for example, here you can take the meristematic cells or the meristem, right? Or you can take vascular cambium, right? Meristem or the vascular cambium. If you take the example of the meristem, what is the function of the meristem? Meristem is nothing but the tip of the plant, right? If these are the leaves, the tip of the plant, the shoot meristem, what is its function, right? It doesn't have any function, but it will divide rapidly so that it will increase the size of the plant. If it is a root meristem, it will increase the length of the root, right? So in the same way, here they regain the division ability, right? They regain the division ability and they lose their function. So these cells are meristematic cells and also the vascular cambium. So here the vascular cambium, vascular system of the plant, what is its function? It has to spread throughout the plant system or the shoot system. So once it is spreading throughout the plant, it will not have any division, right? It will not have any function, but it will divide so that it will go and spread to all the branches, leaves, everything, all the part of the body. It has to get spread, right? So here these are called as dedifferentiated cells. The division ability is regained. Okay, the division ability is regained, but the function is lost, right? Now they don't do any function. Their function is only to, in case of meristem, it has to divide and it has to increase the shoot system. And if in case of root meristem, again the meristem has to divide and increase the root system. In the same way, the vascular system also, it has to go and spread to all the branches and all the parts of the plant body, right? They don't have any function, but their function, their property is only to divide. Okay, so that is called as dedifferentiation or dedifferentiated cells. Okay, next coming to the redifferentiated cells. Now the dedifferentiated cells, the next step is redifferentiation, right? So that means what? Here the division ability is permanently lost. That means here the plant cells has attained the maturity, right? The matured cells need not divide. In case of organs, right? If you take a kidney, liver, everything, from the childhood they keep on increasing their size. After the maturity, the size of the organs is same for throughout the life. They don't increase in their size for the years, right? So once they attain, in the same way here also, all the plant parts, right, they attain the particular size till their maturity. After maturity, here you can see the matured cells with specific function, right, they will then again lose the ability to divide. Okay, they don't divide once the organ is completely formed, right? So now the vascular system, now when it is at a dedifferentiated stage, it will go divide and spread to all parts of the plant body once it is dividing, right? So in case you can take the xylem and phloem vessels, right? This is the plant trunk, right? You have xylem vessels and the phloem vessels. What is their function? In order to transport water and food, right? Your xylem and the phloem. Xylem and the phloem vessels. So here now you can see the xylem, right, which is transporting the water, and you can see the phloem which is transporting the food, right? Here at this stage now all these cells, xylem vessels and the phloem vessels, are matured cells. So they need not divide, but what they do, they perform their function throughout the life. Is that clear? In the same way, here the dedifferentiation or the redifferentiation or undifferentiation or again redifferentiation, all these are mainly important for the plant to survive, right? So first it is the differentiated cell which you are culturing to form a callus, where the callus is undifferentiated mass of cells. Again, the undifferentiated mass of cells are differentiating in order to attain certain function like shoot system with photosynthesis, root system for absorption. Then after that, all the plant parts has to be developed. In that case, again dedifferentiation occurs, right, where the division ability will be again regained, right? After that, again at this stage of maturity of the cells, right, when the cells get matured, they stop dividing and they perform their function. So I think you have known the difference between dedifferentiation, redifferentiation, right, everything. If you have any doubts, again put it in comments. Thank you.